So it's now my pleasure to introduce Janet Woodcock. Janet joined the FDA in 1986 and has had many roles at the FDA, but she is best known as the innovative director of the Center for Drug Evaluation and Research, CEDAR, and for being a total advocate for evidence-based development of therapeutics and the application of contemporary research technologies and methodologies to drug development scientists. She has been a supporter of FDA collaborative research projects with academic scientists and has been a great supporter of our CERSI and this summit for several years, and hopefully several years to come. She's currently on detail in Operation Warp Speed and is a principal advisor to the commission. So Janet, please go ahead.
Thanks very much. So many of the speakers have brought forth the issue of what the performance of our whole biomedical research system during the pandemic. And actually our forthcoming panel is about infectious disease. So I would really like to talk about the performance of the global clinical trial ecosystem in a pandemic and how we did on therapeutic development. You already heard that the biomedical research establishment actually responded incredibly and of course developed vaccines and other specific therapeutics. But how about the clinical trial, the evidence generation system? How did we do? And what are the lessons learned, both for infectious disease and for general therapeutic development? Next slide. Cool. If you think about it, what should be the strategy for therapeutics when you have a new, previously unknown pandemic disease that lacks any therapies? How, in the ideal world, how would one proceed? Of course, the ultimate goal, if you have an infectious agent, is development of safe and effective vaccines to eradicate the spread. But this can take time, both to get them developed, and I think we were really lucky this time, but also to get everyone globally vaccinated. And in the interim, public health measures to minimize spread can be instituted, but they are not going to be completely effective, and they may have severe economic and social consequences, as we have seen. And therapeutics can sometimes be developed more rapidly, can help manage such disease until the other measures are effective, and also for residual cases. So let's use the development of therapeutics for COVID-19 disease as an example. Next slide. So there are two strategies you could really pursue when you have this explosive emergence of a pandemic disease. Repurposed therapeutics are first, and because they can be moved rapidly into efficacy trials, those that are on the market and those that are close to the market. So we saw that happening, and the rationale for clinical testing in this case included in vitro screening results for various antivirals that have been developed for other viruses, anecdotal experiences, case series, and then mechanistic reasoning, say for immunomodulators and in some cases for antiviral activity. A huge number of candidates were identified out of this screening effort. And so then it was left to the clinical trial enterprise to sort through and figure out which ones actually would be useful. There were small molecule direct antivirals that were either approved, such as ritonavir, lopinavir, or in development, such as remdesivir or favipiravir. And there were many, many screening generated compounds that weren't necessarily antiviral agents with putative antiviral activity. And then there were immunomodulatory agents, both approved or in late stage development. Now, if you have an infectious pandemic, infectious agent specific therapeutic development would be expected to take a long time in general. Small molecules, as you know, are really not in the clinic yet because of their development time preclinical. Passive immunity, though, could be brought to bear early. And so convalescent plasma, hyperimmune globulin, and specific monoclonal antibodies are all now in clinical testing, and there are 3 emergency use authorizations that have been granted by the FDA for passive immune approaches. Next slide. So how would you do development programs for the repurposed therapeutics in the ideal world? Well, the ideal thing to do really is generate actionable evidence as quickly as possible. In other words, it could be confidently incorporated into the standard of care immediately after the conduct of the trial. So what actually happened? Screening trials were put up, such as I-SPY, COVID, and BET, the big effects trial. And these were put up in the late stage of disease where there's a very high event rate. And so screening for activity was really feasible. There were serial aimed to be definitive evaluations. So it's sort of ACT is sort of a platform trial. And so they looked at remdesivir and then baricitinib and subsequent. Others put forth large-scale pragmatic trials, such as recovery and solidarity. And in those, because they were highly pragmatic, you really needed to use well-established agents with known safety profiles, so you wouldn't have to do a lot of surveillance. And then there were other platform trials, the ACTIVE series that's ongoing at NIH, and then REMAP-CAP that just read out on one of its arms on the IL-6 inhibitors. And then company-sponsored efficacy trials, which also were IL-6 inhibitors and others. But then there was a very large category of just do a trial. And 95% of clinical trial efforts globally and in the United States on our data were of the last type. Next slide. So we at FDA constructed a data lake looking at all registered therapeutic trials globally, excluding traditional medicine trials. And we found 2610 trials of repurposed agents as of 12/20 registered globally. We evaluated if they had a randomized design and if the planned study power met what we devised as arbitrary but highly reasonable and conservative estimates for having adequate power to actually deliver an answer. Only 5% of experimental trial arms met these criteria, and the same was with trials that were conducted at least in part in the US, 5%. Situation is actually much worse than this because many of the trials that had putative adequate power had poor enrollment. And almost all definitive data have come from platform trials or company-sponsored trials. And the latest, of course, being REMAP-CAP. Next. So there were few trials that were designed to change the standard of care in a pandemic. I think the screening platform trials are a reasonable approach where, as I said, in late stage disease, where you look for big effects, get the answer quickly. But most trials were duplicative. They were small trials and they were all over the place. So if you look at this small table, I have a number of the agents, the number of adequate trial arms. Convalescent plasma, we had about 7 adequately powered randomized trials and 194 other trial arms of convalescent plasma. And likewise, as you look down the list. Next slide. So there were additional problems that we identified. I was involved in setting up the active trials in the US, and but these problems with the ecosystem were encountered across the board. There was a concentration of sites at major medical centers, and this is particularly for government-sponsored trials. And they were competition for patients, study personnel, and time at these limited sites, and also for industry at the CRO-supported sites. There were delays due to contracting, local IRB requirements, lack of PPE, and testing. And the result of this, it gets back to some things that have been said earlier, there was no ability for many of the communities to participate in this research. There was lack of representativeness in these trials. And It was also compounded by the desire of investigators to each do their own trial, part of which is powered, I think, by academic promotion and tenure standards. Next slide. So the controversies that we have right now, we still don't know about the treatment effect of the number of the repurposed drugs, and we've gotten contradictory results in different trials, some smaller than others. Hopefully the larger trials are going to be more definitive. The effect of some widely used therapeutics is still not clear at all, such as convalescent plasma, although it's been used hundreds of thousands of units. There's been slow uptake of the monoclonal antibodies, which likely, very likely, can keep people out of the hospital and protect our overburdened hospital systems. But the data are not definitive. And many claims for effectiveness of various repurposed drugs, and we have proponents of many different kinds of drugs, have gone unanswered because efficient screening and definitive trials are not available, despite the fact that we have hundreds of thousands of affected patients in the United States alone. Next slide. So what should we have done potentially? And we can talk about this a little in the panel, perhaps. For repurposed drugs, I would advise we randomize the first patient. If there's ever a time to do this, it is in a situation such as we have endured. Set up pragmatic screening trials for relatively safe agents. For example, we have a lot of claims about oral outpatient drugs for people with mild to moderate COVID. I wish I had set up a pragmatic, very simple trial for drugs that are not too toxic so they could be screened. You have to realize, though, this would take thousands of people. It isn't like we could do a screening trial of 50 people for this endpoint. Set up definitive efficacy trials for anything exiting the screening trials, either a seamless transition, phase 2, 3, or separate trial, and aim for greater power than what you calculated. Because even the drugs we have met their primary endpoint, we still have controversy and questions about them. And we certainly had enough patients to get better endpoints and better narrow confidence limits on the results. Next one. So what about pragmatic trials versus intensive observation and data collection? We need both. It's desirable to have some planning about the balance. Intensive trials do contribute to scientific understanding. Pragmatic trials are designed to yield single or few definitive answers. There was really no conversation across the clinical trial ecosystem about these matters. The design appeared to rest on the philosophy of the investigator group. We need to plan to create an evidence generation machine that can rapidly generate the knowledge we need if we get in another situation like this. Next slide. Can we do better next time? Well, we're going to have to proactively extend research support into the community. And Kathy was talking about this with regard to the health records, but we also need support and training and research. We need to bring the community into the research ecosystem. Have a plan for coordinated research strategy for the next emergency. Identify sites capable of conducting pragmatic trials, maybe not registration trials for new agents, but pragmatic trials. For federally funded trials, I have some suggestions here of things that could have really streamlined matters quite a bit. And for industry-sponsored trials, FDA might consider triaging criteria to move those platform trials most quickly and discourage perhaps a little bit these one-off kind of trials. Next slide, that's last. So the clinical trial ecosystem response to the pandemic was suboptimal. I think we would all have to agree to that. We will publish these results I have been talking about. Platform trials yielded most or almost all the information, followed by industry-sponsored trials. Most patients were not able to participate in research as usual. and we can and we should do better so we can learn more faster. Thank you very much.
Thank you, Janet. That was great. And we do have some time for questions, and I see some written here. So I'm going to read some of them to you and then have you answer them. So this one is from Rob Califf. I would like for Janet to say what she would do if she were in charge. How do you go, Janet?
Well, I think that the first thing is to have a plan for if I were in charge of this pandemic when it started. I think the best thing to have done would be have a plan, work with the international partners, set up large-scale trials, iterate as information was gathered, really get the community involved in a lot of the repurposed agents. I really regret, as I said, not setting up a outpatient, pragmatic, simple trial to see if any of these oral agents that people are promoting could actually prevent progression and hospitalization, for example. That could have been done out in the community. There are many things that we could have done, but planning and having a more consolidated approach with consolidated objectives, I think would have been much better than the way it played out.
Rolled out, yes. Okay, another question from Peggy McLaughlin. I would like to ask Dr. Woodcock if she would join me in initiating legislation to change the IRS rules around reporting and taxes due for patient reimbursements for clinical trials. I have a call with Congresswoman Jackie Speier. I don't know what you can say there.
Yes, well, I can't, of course, lobby or participate in lobbying of Congress, but we have to recognize that there are problems with our clinical trial. Our approach to clinical trials in the patients is very, very much protective and concerned about exploitation. And there are always two sides to every coin. We don't want to disincentivize people. There are many people arguing that we don't provide enough incentives for people to participate in research and that would be better for them and for people with their disease, for example.
All right, great. Another question, this one's from Kuldev Singh, one of the organizers gave his talk earlier. What can be done to increase the representativeness of U.S. populations in trials for repurposed drugs? What can we do there?
Well, this requires the ecosystem to shift generally, and it can't be done like in the minute, okay? So the different groups such as NIH, industry, and others need to reach out to communities, work with those communities, gain trust, train investigators in those communities, for example, federally funded health centers and so forth, other types of settings, and help them participate in the research enterprise. You can't do it in the middle of a pandemic, or it's extremely difficult to do all those things in the middle of a pandemic. So what we need to do now, I think, is mindfully lay the foundation for investigators in the community to participate in clinical research and be very serious about that, both industry and academia.
Okay, that would be great. Also, another question, what kind of sequencing tools are these trials using? Can innovations in the sequencing methods help with precision medicine? Could that be helpful?
Yes, well, what we discovered with COVID, and we're still learning with the disease COVID-19, all right, is it has phases. And perhaps not all people experience the disease in the same way, and there are different cadences. And for example, we don't have biomarkers, though, now that really illuminate when we should do what. But it does appear we have a period of viral replication. A lot of people's innate immune system control that. They're probably the asymptomatic spreaders, right? So they knock down the virus. Then we have people who the virus replicates more in, but eventually their immune system, their humoral immune system, or their more advanced immune system eliminates that virus. We have other people who the virus keeps replicating. Some of them are not making a a really strong immune response that can control the virus. Other people seem to have other factors. They may have an antibody response, but somehow they're not clearing the virus. And then at some point in this, they transition to a hyperinflammatory state where their immune system appears to be worse than the disease, right?
Yeah.
So if you treat people with steroids early, you're probably going to, there was a trial of inhaled steroids where they showed that people got more pneumonia in the inhaled steroid arm, right? Because they did it early. So if you treat people too early with to suppress the immune system, you may cause the virus to explode. If you treat just with antivirals late, you're missing the main show then. But we don't know how to discriminate amongst those people and what the timing might be. We have very crude measures. So I think the trials, though, as far as efficacy, of course, they're still mainly looking at population means because they're not big enough for the subgroups, right? And that's a problem. A recovery is probably big enough, right? But most of them are not big enough to sort this out. So, but a lot of them are using adaptive designs. They're using seamless phase 2-3. The active trials are doing that and so forth. So you have a screening phase, you can stop for futility, you can readjust sample and so forth, and then you move on without stopping unless you have to have a pause because it's a new molecular entity and you want to be sure of safety. So innovation in those regards are being done, but I would argue that we aren't using enough brute force power to look at subgroups, and this disease is a lot more complicated than some diseases.
It's true. A couple more questions, Janet. So one from Rinko Ghosh. Do COVID therapeutic trials in 2021 need to evolve to account for a major impact of vaccines on the population? And how do we do that?
Yes. Well, we hope we will soon have a major impact of vaccines, but I'm afraid that may be a few months in coming. I think we're still going to be seeing a lot of infections for the next couple months. We're already setting up studies to look at the interaction of vaccination and, say, monoclonal. Say you get vaccinated, you still get sick. Right now, the recommendation is you should still get a monoclonal, right? What if you have had a monoclonal, say, for prophylaxis, or you were sick and you got a monoclonal, then when should you get vaccinated? So those studies are being set up because we do need to answer those kind of questions.
Okay, great. One last question, I think, before we segue over to the panel. So somebody's asking if you have anything to say about trials on the diagnostic side. This is Michael L. Antibody tests, COVID mouth, no swabs, rapid tests. What lessons have we learned on the diagnostic side?
Yeah, I wish I could answer, but I can't. I have not had the time really to educate myself fully on that matter. Clearly, that's the biomarkers, the diagnosis, critically important, and simply diagnosing the disease has been a lot of focus. But obviously, we would like to make that a lot easier, a lot quicker. That's been a huge impediment to the trials. So I believe the technology will undoubtedly get there, but I can't answer that question fully. I will say that, of course, another emerging issue that we'll all have to deal with is the emergence of variants with so much virus replicating and so many humans all around the world and maybe bouncing back and forth from animal hosts. That's a real risk, and there already are efforts being set up to look at that. So that's another issue that we're going to have to keep a really close eye on.
Okay, great. All right, Janet, thank you very much for your keynote talk. And then I guess you're going to be on the next panel, so I will sign off here. Thank you.
Thank you.